My name is James Ehlers, and I've been working with Dr. Coates in the biomechanics department here at the University of Utah. And we've been working in collaboration with the Moran Eye Center to examine the dynamic rheological properties of mouse vitreous. And to give some background knowledge, post-vitreal detachment can cause vision loss if liquefaction occurs early. Collagen 9 is suspected to contribute to the separation of vitreal collagen fibers and regulate vitreal liquefaction. So it's the goal of this experiment to examine the material properties of mouse vitreous and compare it to those of knockout mice, where the gene for collagen 9 has been removed. And because of the nature of mouse eyes being so small, their vitreous is only 4.4 microliters, and that meant that we needed to use rheological experimentation that's not common in industry. So what we chose to do was to use a parallel plate geometry and a 50 micrometer gap height in order to analyze a sample so small. And then we compared that to a gap height of 400 micrometers and one of 800 micrometers as well. And this was done using both smooth plates as well as sandpaper plates as well. And the sandpaper was just to form a layer that would give it more grip to prevent a boundary layer from forming. So that sandpaper we chose is in figure 1, where we've cut out 10 millimeters diameter circles that will be attached to the rheometer head that's 3D printed, as you can see in figure 2. And that will just be done using the adhesive backing from the sandpaper. And the validation material that we chose was a polystyrene toluene melt, as 0.6 grams of polystyrene per milliliter of toluene. And that was mixed continually for about 2 hours before experimentation to ensure it was thoroughly mixed. But the first test that was performed was a strain sweep, and that was to determine the linear viscoelastic region of the sample. And that can be seen in figure 3, where there's this gentle curve upward, and then the storage and loss modulus both seem to converge and flatten out into a linear pattern before diverging and curving again. And so when we found the linear viscoelastic regions of all the samples, both with and without sandpaper and at multiple gap heights, and averaged those in figure 4, we found that there was a linear viscoelastic strain of about 2.51%. And so that will be used in the future when we complete our frequency sweeps as well. But the next test performed was a stepped flow test, and that was in order to determine the viscosity of the sample. And a typical sample that we would expect is shown in figure 5, and that was one of the results of this experiment, and is probably the most clear sample, where we have a Newtonian region just to the left of 1 inverse second, and then a trail linear offward to the right. And so when we examined our data and averaged it at each different gap height, we got the results in figure 6. And what we found is that most of the samples were within the same region and exhibited similar patterns, but the 50 micrometer gap heights for both sandpapered and smooth heads seemed to diverge from the rest of the results. And that sent up some red flags for us, because the viscosity is not supposed to be a function of the gap height. So that meant that some error had been entered into our experiment somewhere. One of the possible sources of error there could be that the geometry of a 3D printed rheometer head is not within the tolerances needed for such a small gap height. So that'll be something that we go and revisit in the future. But in order to proceed with the experiment, we need to go back and define those sources of error and eliminate those before proceeding. But once those are eliminated and diagnosed, we can move forward and complete the frequency test to complete the validation experiment.